Good morning, dear students. Today we are going to study about the male reproductive system. As you know, a reproductive system or genital system is a system of organs within an organism which works together for the purpose of reproduction. The main functions of a reproductive system are to develop secondary sexual characters, then produce gametes, produce hormones that help in the regulation of the reproductive system, as well as produce an offspring by fertilization. So male reproductive system, the course outcomes are to study or to understand the different organs as well as glands involved in the working of the male reproductive system, as well as to correlate the anatomical structures with their physiological, endocrine and biochemical functions. So the learning objectives would be, we have 4 learning objectives in the current topic. That is, first one is to identify the primary, accessory and supportive reproductive structures involved in the male system, then to map the precise anatomical pathway of sperm from production to delivery, to analyze the biochemical composition and clinical profile of semen, as well as to detail the different stages of spermatogenesis and correlate its hormonal regulation via the HPG axis, that is the hypothalamus pituitary gonadal axis. It mainly consists of the primary sex organs, which are the gonads, that is the testes. These are responsible for the production of sperm and critical androgenic hormones, that is the testosterone. Then we have the accessory sex organs, which mainly consist of the ducts like epididymis, vas deferens and urethra, which is responsible for storage and transport of sperm. Then we have the accessory sex glands, that is the seminal vesicles, prostate gland and the bulbourethral gland, which are responsible for semen production that helps in the nourishment of sperms and also acts as a medium for the transport of sperm. Then we have the supporting structures, that is the scrotum and penis, which are anatomical structures that assist in the temperature regulation, delivery and joining of gametes. So as we know, the testes, which are the main male gonads, they are about 5 cm in length, 2.5 cm in diameter and weigh about 10 to 15 gram. They are covered by a sac-like structure known as scrotum, and the male testis descends to scrotum via inguinal canal at the 7th month of fetal development. Now, moving on to the histology of testis, the testis is covered by 2 protective layers, which is the tunica vaginalis, that is the outer serous membrane, and the tunica albuginea, which is the inner dense connective tissue. Now, the tunica albuginea extends inwards to form septums, which divides the, as we can see here, which divides the testis into different series of compartments. Each compartment is known as a lobule, and each testis consists of about 200 to 300 distinct lobules. Inside the lobule, we can see the presence of 2 to 3 tightly coiled seminiferous tubules, which are the site for sperm production or spermatogenesis. So moving on to the seminiferous tubules, mainly consists of 2 types of cells, that is the spermatogenic cells and the Sertoli or sustentacular cells. The spermatogenic cells are the stem cells that actively differentiate to produce a mature sperm, while the Sertoli or sustentacular cells extend from the basement membrane to the lumen. They are present neighboring to the spermatogenic cells, and they act as a nourishing agent for the spermatogenic cells. They form the vital blood testis barrier, nourish the developing sperms, secrete fluid which is required for the transport of sperms, and also help in synthesizing the androgen binding protein, wherein the male sex hormone binds and is responsible for the different functions of testosterone. The Sertoli cells also mediate the regulation of FSH and testosterone action. Neighboring to the seminiferous tubules or located in clusters in between the adjacent tubules are the Leydig cells, which are also known as the interstitial cells. These cells synthesize and secrete. So the scrotum, as we already know, is a sac-like system that helps in protecting the testis or covering the testis from external environment. It maintains an environment of 2 to 3 degrees Celsius below the core body temperature, which is essentially for viability of sperms. It mainly consists of two types of muscles, that is a dartos muscle and cremaster muscle, which contract during cold or arousal to absorb body heat and relaxes during warmth. Then moving on to the penis, we know that it is a supporting structure that acts as a passageway for both semen and urine. The penis consists of it has a cylindrical shape and consists of three parts, that is a root, body and glans penis. The core of the penis contains three erectile tissues, that is two corpora cavernosa layers and the corpus spongiosum. Now the root part, which includes the bulb of penis and crura, it is a proximal portion. The bulb of penis consists of corpus spongiosum, while the crura of penis consists of corpora cavernosa. Now the body part, which is the main part, it consists of three cylindrical erectile tissues, that is two dorsolateral corpora cavernosa and one medial corpus spongiosum. These muscle masses are covered by skin, fascia, tunica albuginea and tunica albuginea. Now the distal part is the glans penis, which is a slightly acorn-shaped enlarged portion of corpus spongiosum. It is surrounded by a loose skin known as prepuce or foreskin. The proximal margin of the glans penis is known as a corona, and the opening in the distal end is the external urethral orifice. So the next topic is the structure of sperm. Sperm is as we already know, sperm is the male gamete which is produced in testis. About 300 million sperms per day is produced by spermatogenesis, which takes place in testis, and the lifespan of a sperm is about up to 48 hours in the female reproductive tract. So the parts of a sperm are mainly head and tail. The head part, the length of a sperm is about 60 mm. The head part is a flattened pointed head that is about 4 to 5 mm long, and it contains a nucleus and acrosome. Nucleus consists of 23 highly condensed chromosomes. That is, it contains the main genetic material from the father's side of the embryo. The acrosome is a cap-like vesicle that contains enzymes like hyaluronidase and protease that help the sperm to penetrate into secondary oocyte and bring about fertilization. Now the tail part helps the sperm to move in the female genital tract, and it mainly consists of four parts, that is the neck, middle piece, principal piece and the end piece. The neck is a constricted region below the head, that is this part. Then it contains centrioles. The centrioles form the microtubules that form the remainder of the tail. Now the middle piece, that is this piece, this is the middle piece. It contains mitochondria which are arranged in a spiral fashion, provides the energy for locomotion of sperm to the site of fertilization and for sperm metabolism. The principal piece, that is this portion, is the longest portion of the tail, and the end piece is the terminal tapering portion of the tail. Now the seminal vesicles. Now moving on to the composition of sperm and seminal fluid, that is mainly the semen. It consists of secretions, as we already learned before. It consists of secretions from the seminal vesicles, prostate gland and the bulbourethral gland. Each secretion from each gland helps for the nutrition of sperm as well as the motility of sperm, and also to maintain an optimal environment for the sperm to remain viable in the female reproductive tract. The seminal vesicles secrete an alkaline viscous fluid that contains fructose, prostaglandins and clotting proteins. Prostate gland secretes a milky, slightly acidic fluid that contains citric acid, acid phosphatase, proteolytic enzymes and seminal plasmin, whereas bulbourethral glands secrete an alkaline fluid that protects the passing sperm by neutralizing the acidic urine in urethra, and it also contains mucose and galactose. Within 5 minutes of ejaculation, semen coagulates due to the clotting factors present, but after 10 to 20 minutes it reliquefies due to prostate specific antigen as well as the proteolytic enzymes. The fructose, citric acid and galactose present helps to provide ATP for the sperm motility, while other factors also help in the nutrition of sperm. Female vagina.